Hey guys, today's notes are going to be on the cell membrane. So we're going to talk a little bit about diffusion and the movement of molecules and all that kind of stuff. The document that you're working on looks like this. The first thing you're going to see on that document is a picture with A, B, and C. And then a chart below A, B, and C, and you're going to tell me as I go through the notes what it is and what is it there for. Feel free to pause the video at any point so that you can type in what you need to type in. I usually do my notes videos kind of fast because there's no interaction with other humans. It's just me and the camera. But so that means that if you need to pause it and go back, then you need to do that so that you can appropriately fill in your note packet. There's a point at the end I will show you that gets a little bit tricky, but we'll get there when we get there. So, not the right button. Present. No, still not the right button. So diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So diffusion is what happens at the cell membrane. Cell membrane surrounds both plant and animal cells. Diffusion is what happens at the cell membrane. The cell membrane determines what can enter or exit the cell, and it determines that based on the size of a molecule. So the example I use in class a lot is in my classroom, without destroying the walls and the whole rigmarole, could you drive a car into my classroom? The answer is no. No, you could not, because the car is too big. The next question then I say is, okay, well, could you, without destroying anything, could you drive a motorcycle into my classroom? And the answer is, yeah, you could. You'd have to drive it down the hallway and in the front door, but you could drive it into my classroom without destroying anything because the motorcycle is small enough to fit through the openings in the room, similar to cells. Okay? So I'll put myself up there for now. We'll see if that lasts. So we're looking at the cell. We're looking at basically the outer edge of a cell. We're taking that circle that we usually draw to describe our cell, and we're just like zooming in on one little section of it. So that's why part of this is marked as outside the cell and part of it is marked as inside the cell. So letter A is pointing to something called proteins. Proteins help things move through the cell membrane. It's the equivalent of saying that if my window is only open this far, then I can't throw a beach ball out of it if the room is the classroom still. But if I open the window the rest of the way, I can throw a beach ball out the window without destroying the classroom. So you can still move molecules through you, just the protein needs to help. It needs to open up a little bit. The second letter, letter B, is the carbohydrates. Carbohydrates are what we call it like ID tags. They basically tell you what kind of cell it is, not you personally, but tell other cells and other things that are outside the cell, molecules and other things that are trying to get into that cell. It gives them information about what is the cell, what kind of cell it is, and what's going to happen when that molecule gets inside. And part C is a lipid bilayer. So you're seeing the lipid bilayer. This right here is a lipid. A lipid, I always say, is kind of like a balloon with two tails. That's one lipid. So the lipids are sort of like floating on top of each other. They're not stuck in space. They float kind of back and forth on top of each other, which makes it so that molecules can pass through them as long as the molecules are small enough. We call it a lipid bilayer because there are two layers of lipids. The lipids are sort of the heads of the lipids, which are like the balloon parts, are repelled from each other, and the tails end up facing each other. And that's how they always end up there. It's a chemical reaction that causes them to be repelled from each other, kind of like when you put oil into water and the oil clumps together and like repels the water. So it's basically called the lipid bilayer because it is two layers, bilayers of lipids floating on top of each other. There are three functions of the cell membrane. It regulates what enters and exits the cell. It provides some protection, not as much as the cell wall, but some protection. And it provides some support. Again, not as much as the cell wall, but it does provide some support. So then there's a couple questions there. It says, it talks about selective permeability or semi-permeable. So to be semi-permeable basically means in non-science terms to be kind of, which is what semi means, kind of, semi. And permeable means like able to be passed through. So a window is not very permeable at all. But a screen in the window, if you open your window and you have a screen in, the screen is semi-permeable. Small things that can fit through the little holes of the screen, those can pass through airflow, pollen, things like that. Some little teeny tiny gnats sometimes and bugs can pass through. But the plants, the larger molecules can't pass through. So it is semi-permeable. That's what it means. So when we say semi-permeable, we mean it chooses what gets in and what does not get into the cell. Molecules that are usually easily passed through the membrane are small molecules. They're also uncharged. So some molecules have charges to them and some don't. It's not super important that you know that. It's more important that you know the small versus large instead of the charged versus uncharged. But it is a thing that the uncharged ones get in easier. And then the molecules that have difficulty getting through are the larger ones and the charged ones. So, move my face again. Diffusion is movement of molecules. So this is all types of movement of molecules through the cell membranes. Movement of molecules from a high concentration to a low concentration. It's kind of like sledding. You start at the top of the hill and you fly down to the bottom of the hill. So you're moving naturally from that high to the low. It's called moving with the concentration gradient. And it does not require a lot of energy. So if you think about sledding, when you are sledding and you fly down the hill, it doesn't really require a lot of energy for you to just fly down the hill. You're going from the high spot to the low spot, and it happens fairly naturally. Once you get yourself kind of scooched to the crest of the hill, you're going. You're not stopping even if you wanted to, if it's a good enough hill. So that's what we mean by with the concentration gradient. A couple of examples of things that diffuse very easily are CO2 and oxygen. So if we were to look at this picture, you can see that there's a high concentration of these dot molecules on the left-hand side and a low concentration of these molecules on the right-hand side. So the molecules are going to move from left to right, okay? In your document that you're taking notes on, you're going to double-click on this picture. And if you use your shape option, which is the circle and the square, and you go down to arrow, you'll be able to put in that right-facing arrow. You don't need to, I think I did 3. You don't need to do 3. You can leave it just like that. If you want to change the color on the inside, you can by hitting this color fill thing, but you don't need to. Okay, so that's how you're going to do that, and then you're going to save and close. And then it will be, but it will be in your notes. I'm not going to keep it in my notes because I don't want it in my original. Okay, so let's see if I get it back. There we go. Yeah, I already did you. Concentration, no energy, CO2, O2, good and beautiful. Okay, next one is osmosis. Osmosis is still a type of diffusion. It's just diffusion of water, so it's a special kind of diffusion. It's still diffusion, so it's still moving from a high concentration to a low concentration. And it's still diffusion, so there's still no energy needed. It's still going from the top of the hill to the bottom of the hill. It happens very naturally. Doesn't need any energy to be pushed behind it. It's kind of like if you've ever been in a lazy river, and the lazy river usually has a current going in one particular direction. And if you sit in your tube, you're just going to go. It doesn't take a whole lot of energy. It's lazy, just like it's called. You just hang out, and it will take you around in the direction that you're supposed to go. If you wanted to get off your tube and go backwards in the wrong direction, then first of all, probably you get yelled at by the park police or the people who are in charge of the water park that you're at. But it's going to take a lot of work, right? You're going to have to kind of sludge backwards. You're going to have to swim against the current and kind of work your way backwards. So it takes a lot more energy. Oops, sorry. I didn't mean to delete those. So that's the opposite of what we're talking about here. This is a high to low. So it's very natural. It happens easily and there's no energy needed. The last one here for this part is called facilitated diffusion. So if you don't know the word facilitated, it really just means helped diffusion. So this is where those protein molecules from the front of your packet and the picture come from. It's still diffusion, so it's still moving from a high concentration to a low concentration. It does, however, now need a helper molecule. It's kind of like I was talking about how you'd have to open the window bigger. You got to make it a little bigger. You need a little helper molecule to open the window bigger, but it can still happen. Something can still pass through the space. It does not need energy. Goodness, it does not need energy. I need coffee, apparently, though. Sorry. Does not need energy. And some things that are a little bit larger, but not too large, that can pass through the membrane with the helper protein are things like calcium, which is Ca, magnesium, which is Mg, and salt, which is actually NaCl, is salt. So, actually, let me go back one. So, now on your page, you're probably seeing, hopefully, Okay, so we're done with osmosis. We did facilitated diffusion. Now you see all these stars separating this from the next section. This next section, it's separated on purpose because this one is basically the opposite of what we've been doing in diffusion. So this one is called active transport, and it is exactly how it sounds. It is active. It takes energy. It's the equivalent of being done on the bottom of the hill at the sledding hill and having to sling your sled over your shoulder and climb your way back up to the top because you want to go again. It's hard work, especially if the snow is really deep. It's tough work. So it's going from a low concentration to a high concentration. This one we call moving against the concentration gradient. It's going uphill. It's going against the current. It's going however you want to think about it. It's hard work. Okay? This one absolutely requires energy in the form of ATP. And one of the things, or a couple of things that will do this are things like sodium, which is what Na stands for. It's a chemical name for sodium. And K is actually potassium, which doesn't make any sense any more than sodium is Na. But somebody somewhere, it makes sense to them, just not me. But these two things, sodium and potassium, are things that often move against the concentration gradient and need energy to be pumped into the cell for whatever reason. Okay, so again, in your picture, you're going to have the same picture again. You're going to double click on this picture. You're going to go to your shape menu with the circle and the square. You're going to go to the drop down where there's arrows, and you're going to think about, all right, well, if this is my high concentration and this is my low concentration, which way are the molecules going to move in active transport? They are, in this case, going to move from low to high. So your arrow is going to point, I guess that would be that way to go along with the fact that my video is backwards. Okay? So molecules will continue to move. That's what that says. Continue to move until they reach a state of what's called dynamic equilibrium. And basically, dynamic equilibrium just means, that the concentrations are equal on both sides of the membrane. It's really important that they do that. That's what they want to do. Molecules naturally want to move. They naturally want to spread out as far as they possibly can. If they are constrained by a barrier, then they can't go any further because they're too big. But they will start to spread out as best they can in whatever space that they are given. That's what they want to do. So let's look at a couple of examples. Oh, before we start this part, I'll look at your note packet. So in your note packet, you notice that you have a chart here. It also says what happens when, and then it says use the pictures below the chart to copy and paste the appropriate after pictures into the chart below. So here's your pictures down here. There should be 6 of them. My friends in the pink class, there might only be 5. I'm really sorry. I fixed it, but it was too late for you. But when we get to the one that's missing, you can just copy it from the chart and move it over. I'll tell you which one that's missing, okay? So you actually can probably figure out which one's missing because you're seeing 6 on the video and there's probably only 5 on your paper. So I apologize for that. But instead of having to draw these, you're just going to right click on them, copy them, and you'll be able to paste them into these after boxes as needed. That's not the right one. Don't put that one there. Okay? But just so you know, then you won't have to draw all of these doodles. Okay? So if we put animal cells in something called a saline solution. Saline solution is about 98% water, which is actually about the same percentage of water that your cells are. Animal cells are about 98% water. The other 2% is like salts and minerals and other things like that. If you have a cell in 98% environment and it's 98% inside, they're equal, right? There's not one that's higher than the other. We're looking for a high concentration versus a low concentration, and these two numbers are equal, which means they're already basically at that dynamic equilibrium place. So over time, nothing's really going to happen. The cell is going to stay the same here, okay? So this one I believe you can copy and paste into your chart from the bottom. I think it's the next one that's missing. It's the plant cell saline that's missing. Okay, so now if we look at a plant cell, same thing, in saline solution, so 98% water, equilibrium, they're already equal in this saline solution. So nothing is going to change. So it's going to stay the same. I think this is the picture that some of my friends in my pink class are missing. So if this is the one that you do not have in your little picture bank below the chart, then go ahead and just copy this picture from the before and paste it over there. No, it's the same picture. It will be fine. And you can, if you want to, you can type in it. It says the cell will stay the same, but it doesn't matter. So now let's say we take our plants and we put them in a salt solution. So now they're only 85% water. Now it's 15% salt. We're looking at the water movement. So our inside of the animal cells are still 98% water, but there's only 85% water outside. So there's a higher concentration inside and a lower concentration outside. So which direction is the water going to want to diffuse? Remember, it wants to diffuse. It wants to spread out from where it's more concentrated into an area where it's less concentrated. So it's going to want to spread out and it's going to diffuse out of the cell. Okay? As a result of diffusing out of the cell itself will actually shrink. Okay? The cell membrane will shrink and it will kind of look a little bit like a deflated balloon, which is the best I could do with this doodle here. So copy and paste that one into the after. We'll be good to go. So for plant cells then, same thing. Salt solution, lower concentration of water outside, higher concentration of water inside. The water is still going to want to diffuse out. The difference with a plant cell is only the cell membrane, which in the plant I've turned it green, only the cell membrane is going to shrink. Only the cell membrane is going to shrink. Okay? The cell wall is going to remain the same. So the cell membrane will shrink. The cell wall remains the same. And you should have this whole picture. I know it's animated separately, but you should have this whole picture in your picture bank below the chart that you should be able to copy and paste it right over. Okay? It's annoying me that that's not too. There we go. It's annoying me it was on 2 lines. Silly Sallies. Okay, so the last solution that we're going to look at here is what's called distilled water. Distilled water is 100% water. It is absolutely pure H2O molecules and nothing else. No salts, no minerals, no nothing. This is not the water you drink. You drink water that's 98% water because we don't want to drink the 100% water, and you're going to see why. So if we are animals, any other animals, our cells are still normally 98% water inside. If we put them in an external environment that is 100% water, the water is going to diffuse in which direction? It's going to move from that high number to the low number. Okay, so it's going to try to diffuse in. What happens to this cell when it diffuses in? It's getting more and more water inside that cell, and it's like a water balloon. What do you think might happen to the cell? The cell will swell. Usually it starts by swelling like a water balloon, and then eventually it explodes. This is no bueno. We don't want this. Oops, sorry. This is no good for our bodies. We don't want our cells to explode. We don't want our other animal cells to explode. This is not a good thing. Okay? So this is why we don't drink distilled water. We drink water that's like 98% water. The water that comes out of your tap, your fridge, your bottled water, it's all like 98% water. There's a little bit of something else in there. And that's usually why, I don't know, some people disagree with me, but I believe that most bottled waters each have a unique taste to them. And I think that's why, because it's the 2%. I feel like Dasani has a totally different taste than Aquafina or Poland Spring or different types of bottled water. I don't drink Nestle bottled water because I think it's gross. I don't know what it is, but they're putting in that 2%. I think it's gross. Okay, so then in plant cells, same concept here. We're still putting our plant cell in distilled water. So we have our normal 98% inside the cell, and our plant cell is 100% in a distilled water environment. So still here, the water is going to want to diffuse into the cell. It's going to want to move from the higher concentration to the lower concentration. It's not a huge difference, but it's still going to want to balance itself out. The difference here is that this cell membrane will swell, just like the other one, just like the animal cell will, but it will only swell as far as the cell wall, because the wall is like that nice, hard, rigid structure. I got the wall right next to me. Nice, hard, rigid structure. It won't swell past there. It will just like fit. It would be like blowing up a balloon in a box. It will fit into the crevices, into the corner, and it will kind of swell out really big, but it won't explode. It's protected by that cell wall. Okay, I think that is the last slide. Yes, it is. So after this, you are going to be doing some practice questions, and we'll be doing a couple of diffusion labs. If you have any questions about diffusion, you can refer back to this video, you can refer back to your notes, or you could always ask a few questions. Have a nice day.